Let us briefly summarize the most important highlights in cytoskeleton. First, let's discuss microfilaments. So they are called microfilaments because they are the thinnest filaments from all the parts of cytoskeleton with a diameter roughly 8 nanometers. And the distribution inside a cell suggests that the microfilaments are organized mostly as the cortical cytoskeleton, reinforcing, maintaining the cell shape, and they are also projecting into various cell projections that might be formed here. Projections and processes, and this would be the cortical cytoskeleton. The basic unit is a globular protein called G-actin, G like globular, which has a tendency to polymerize into fibers. That's the fibrillar actin or F-actin. And the double helix, or two F-actin molecules organized as two helical molecules wrapped around each other form the actin microfilaments. So it's composed of two double helices of the F-actin. They can grow or polymerize or depolymerize, and they can be reinforced by various cross-links. So they reinforce cell processes such as microvilli or stereocilia. They also maintain the cell shape, and they have an important function in mechanotransduction, which means they are transmitting forces, external forces, into the cell, because cell can react on mechanical stimuli. So they transmit mechanical forces, tensions, pressure, etc. They maintain the cell shape. Okay, they control also the flow of cytoplasm. And arrangement of organelles. Other class of cytoskeleton could be intermediate, filaments. They are somehow thicker, ranging between 10 to 12 nanometers. They have a little bit different distribution inside a cell. So this is the nucleus. They are radiating to all directions. And we got various types of intermediate filaments that are often tissue specific, which is used in diagnostic histopathology. So, for example, cytokeratins are intermediate filaments typical in epithelia. Another intermediate filament is vimentin. Vimentin is is present in cells of mesenchymal origin. Which includes connective tissue cells, and also smooth muscle cells. For example, we got desmin, which is a typical intermediate filaments of the muscle tissue. Right? We got neurofilaments present in neurons. We got GFAP, which stands for glial fibrillar acidic protein, which is in neuroglia. Neuroglia are supporting cells in the nervous system. We got nestin, being present in some undifferentiated cells. We got lamins that are anchoring chromatin to the nuclear membrane. And more. So the intermediate filaments are connecting organelles with cell membrane. They are relatively stable. And they are anchoring to desmosomes. So the desmosomes are one of the cell-to-cell junctions that will be discussed in epithelial tissues. So that will be desmosome. It's a type of intercellular junction. Another class of cytoskeleton are microtubules. They are organized around a structure called centrosome, which are two hollow cylinders perpendicular to each other, and the centrosomes serve as a organizing center from which the microtubules radiate into other compartments of the cell. So they are the thickest structures with a diameter of 25 nanometers. They are hollow, rigid, rigid tubules. Organized around the centrosome. And they are made of alpha and beta subunits of tubulin, alpha and beta subunits, or tubulin subunits, that form a heterodimer made of these two subunits that has a tendency to polymerize into protofilaments. And these protofilaments are organized into microtubules. So usually there are 13 protofilaments, 1, 2, 3, 4, 5, 7, 8, 9, 10, 11, 12, 13. So there are 13 protofilaments. that form the microtubule. There's a plus end where the structure can grow, polymerize, and there's a minus end with the opposite process, where the microtubule can depolymerize, therefore growing or not. The microtubules are sometimes forming doublets or triplets. A typical arrangement would be a microtubular doublet, where we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments forming a complete microtubule, and there is adjacent one made of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to 11 microtubule filaments. Okay, and this is called a doublet, microtubular doublet. They are present in the mitotic spindle, they are in the kinocelia or in the flagellum. Kinocelia could be found, for example, in the oviduct or in the epithelium of nasopharynx, nasal cavity, trachea or bronchi. A flagellum is responsible for the sperm motility. In neurons, they are called neurotubules, and they are responsible for axonal transport, which runs even through the very long processes of neurons. And they form tracks for vesicular transport, where various substances are transported within the cell or to the cell membrane before exocytosis. It's called vesicular transport, and the tracks are the microtubules, and the molecular motors such as dynein or kinesin are moving the vesicles along these tracks. We need to mention also the centrosome. More briefly, it's made of two centrioles, which are hollow cylinders occurring perpendicularly to each other, around which the microtubules are organized. Pair of centrioles that are perpendicular to each other, and each centriole is made of 9 triplets of microtubules. 1, 2, 3, 4, 5, 6, 7, 8, 9, and we got a triplet A, B, C, and so on. And they are connected, they are stabilized, and there are some radial spokes. Okay, so there are 9 triplets, 9 microtubule triplets. There are microtubule A, B, C. So this would be one of the centrioles. Okay, this is a centriole. And the centrosome contains proteins regulating the growth and arrangement of the microtubules, right? So it's a center organizing the microtubules. During the interphase, they're usually close to the cell nucleus. In some cells, in cells with kinocilia or flagellum, it can divide, and it can become the basal body of these structures of kinocilia or flagellum that anchors these structures into the periphery of the cell. And during the mitosis, it organizes the mitotic spindle that is responsible for distributing the chromosomes into the daughter cells during the cell division.